Eu gostaria de convidar a Cristina, da MERK, ela vai falar daqui a pouquinho, eu já vi, tá bom? Estou esperando. Enquanto isso, eu vou contar umas piadas. Então, o pessoal da MERK, né, colaborou com a gente para poder construir esse evento. Então, eles vão ter alguns minutos para mostrar alguns dos produtos deles, né, que a gente usa com bastante frequência. Então, o pessoal lá em cima está colocando a apresentação, e aí a Cristina, da MERK, vai falar com vocês sobre os produtos, tá bom? Até lá, tenham paciência.
A Sigma pode?
Falei 1h, 13 horas.
Tá bom. É que estava marcado inicialmente para 11h45.
Não, mas eu fui ali fora, falei que era 13 horas.
Ah, tá bem. Achei que ia ser quando as pessoas retornassem, não tem problema. Foi a apresentação mais breve que eu já fiz, obrigada.
Deixa eu te explicar.
Não tem problema, entendo do horário.
Não, peraí. Nós estamos com a agenda super complicada. E a questão... Não, mas deixa eu me explicar, ué. Aí eu falei, tal, ia cair na hora do almoço, eu falei para o pessoal, deixa eles começarem às 13 horas. E eu, inclusive, pedi para várias pessoas chegarem aqui 13 horas. Agora, eu não posso abrir mão desse.
Não tem problema. Como comentaram com a gente às 13, a gente entendeu que era no retorno, a gente até perguntou lá fora da Beth. Ela falou assim, quando o pessoal retornar, então a gente ficou esperando. A gente ficou esperando o fórum, mas não tem problema. Não tem importância, professor.
Boa tarde a todos. Hoje eu vou apresentar o resultado de um trabalho realizado nas últimas semanas.
Prezados convidados, colegas, amigos e, em especial, nosso querido e inspirador professor Ricardo Tostes Gazinelli. Hoje, nossos corações se enchem de emoção, orgulho e profunda gratidão. Celebramos não apenas uma brilhante trajetória científica, mas a história viva de um professor que, com generosidade e paixão, transformou vidas. Um mestre que inspirou gerações, cultivou ideias, guiou descobertas e formou não só pesquisadores, mas também seres humanos mais curiosos, éticos e comprometidos com o conhecimento. Celebramos, sobretudo, a jornada de um cientista cuja presença moldou e continua a moldar os caminhos da imunologia no Brasil e no mundo.
Eu fico muito contente de participar dessa homenagem para o Ricardo. Eu acho que o Ricardo foi uma pessoa extremamente importante para a minha trajetória. Apesar de eu nunca ter sido formalmente aluno do Ricardo, eu tive uma interação muito grande com o Ricardo desde o meu doutoramento, quando eu fazia pesquisa com o Rabinovich, e o Ricardo na época estava aparecendo os receptores TOL, a gente colaborou, então tive artigo de primeiro autor que o Ricardo é o último. Foi uma cooperação muito legal, ia para Belo Horizonte muito, conversava com os alunos, então me sinto parte aí do grupo de pesquisadores que foi diretamente influenciado pelo Ricardo.
São 30 anos de convivência e o Gazinelli sempre foi para mim um exemplo, uma pessoa em que eu mirei, né, e quis seguir mesmo os passos da vida acadêmica, de pesquisa.
Eu acho que ao longo desses vários anos que eu tenho trabalhado junto com o Ricardo, especialmente nos últimos 10 anos trabalhando juntos no CTVacinas, a gente percebe que o Ricardo é uma pessoa que tem uma capacidade absurda, incrível de conseguir agregar pessoas, pessoas de diferentes especialidades, diferentes personalidades, em torno de um projeto único e um projeto desafiador. Então, isso é raro de encontrar nas pessoas e o Ricardo tem essa capacidade.
O professor Ricardo nasceu em Nova York em 1960, mas foi em solo mineiro que suas raízes científicas realmente floresceram.
Ricardo, eu também quero prestar minha carinhosa homenagem a você, reconhecendo uma carreira que você fez com muito esforço, mas que foi muito bem sucedida. E eu quero lembrar um caso de você pequenininho, com uns 4 anos, tomando banho, e que me perguntou por que a gente consegue enfiar a mão na água e não consegue enfiar a mão na mesa. Aquilo mostrava uma curiosidade que já não era uma coisa tão comum.
Sua carreira científica teve início ao ingressar na graduação, onde sua curiosidade e dedicação o levaram desde cedo aos caminhos da pesquisa. Sua iniciação científica foi orientada pelo Dr. Enio Cardillo e, posteriormente, pelo Dr. Tássio Moraes Santos. Graduou-se em Medicina Veterinária pelo UFMG em 1985 e, com notável precocidade, concluiu doutorado em Bioquímica e Imunologia pela mesma universidade em 1989, sob a orientação do Dr. Giovanni Gazinelli e co-orientação do Dr. Zygmunt Brenner, 2 grandes nomes da ciência brasileira.
É uma enorme alegria participar dessa homenagem ao Ricardo, um pesquisador brilhante, ético e altamente comprometido com a ciência de boa qualidade.
Oi pessoal, eu estou extremamente honrada de fazer parte dos 30 anos de carreira do professor Ricardo e de poder estar aqui hoje falando um pouquinho sobre ele. Trabalhar com o professor Ricardo é extremamente gratificante e empolgante. Ele é um mentor de verdade, que além de ter contribuído diretamente para a ciência brasileira, ele também contribuiu na formação de diversos alunos e pesquisadores. Ele sempre nos ensina a fazer uma ciência de qualidade, a orientar novos alunos, escrever projetos.
Bom, falar do Ricardo para mim é muito fácil. Ele sempre foi uma grande inspiração para mim, desde o início da minha jornada científica. Inclusive, ele fez parte da minha banca de doutorado, foi a minha primeira escolha para compor essa banca de doutorado.
Eu vir aqui mesmo, apesar de eu não poder estar aí presencialmente, queria te parabenizar por esses 30 anos de imunologia, 30 anos brilhantes, à frente, sempre à frente do seu tempo, uma imunologia de ponta, de liderança não só nacional como internacional. Parabéns, você realmente tem feito muita diferença no nosso campo de atuação.
Oi Ricardo, bom, infelizmente não vou poder estar aí com vocês nessa celebração desse feito incrível, 30 anos, né, de laboratório, vários laboratórios de sucesso, mas não poderia deixar de desejar que os próximos 30 anos sejam tão brilhantes quanto esses primeiros, que você continue sendo esse farol que nos mostra o caminho de uma ciência ética de qualidade.
Ainda durante o doutorado, desenvolveu a parte prática de sua pesquisa no Instituto René Rachou, Fiocruz-Minas, onde iniciou sua atuação em pesquisa em 1986. Em 1988, deu mais um passo em sua formação internacional ao participar do prestigiado curso de verão de Biologia do Parasitismo no laboratório Woods Hole, nos Estados Unidos, ampliando ainda mais seus horizontes científicos. Em 1994, deu um salto internacional ao realizar seu pós-doutorado em imunologia celular no National Institute of Health, NIH, nos Estados Unidos, sob orientação do Dr. Alan Scher.
Hello, Ricardo. As your colleague, friend, and former mentor, it's really a special pleasure to congratulate you on 30 years of groundbreaking biomedical research. We personally benefited from your enormous scientific talent when in the 1990s you joined my lab at the NIH as a postdoctoral fellow, launching a new program on the immunology of toxoplasma infection, which at the time was a major scourge of AIDS patients. Amongst other important discoveries, your work uncovered a key role for interleukin-10 in preventing infection-induced immunopathology and set the stage for years of research by a generation of young scientists who later followed you in my lab. Since returning to Brazil, you have built a world-class laboratory and now research center and extended the scope of your research to malaria, viral infection, and vaccinology, while mentoring your own cadre of highly successful investigators. At the same time, both your science and leadership have been international in scope, building strong ties with foreign institutions, and most notably the University of Massachusetts, where you maintained a second laboratory for many years. Importantly, throughout this highly productive 30-year period, you have remained a loyal and generous colleague for all of us who have had the special pleasure of working with you. Knowing you, Ricardo, 30 years is likely to be just a passing benchmark in what will be a long and remarkably creative and innovative career. So on behalf of myself and your long-standing colleagues at the NIH, parabéns. We hope that you're enjoying today's special and well-deserved tribute to you and your important work.
Sabia de Ricardo muito antes de conhecê-lo, porque seu pai Ramayana, com quem tive o privilégio de compartilhar várias comissões federais, sempre falava do seu filho e do sucesso que ele tinha nas suas pesquisas no NIH. Mas como era seu pai, eu levava leve. Mas fiquei encantado quando conheci o Ricardo, pelo seu conhecimento científico, pela profundidade com que ele levava todas as questões científicas, com a sua capacidade criativa e também a capacidade de gerar grupos que funcionavam muito bem tanto aqui como na Universidade de Massachusetts, nos Estados Unidos.
Essa experiência o conectou ao que havia de mais avançado na ciência global e serviu de alicerce para uma carreira que viria a ser referência internacional. Foi sob a supervisão do Dr. Alan Scher que Gazinelli desvendou o importante papel da IL-10 na regulação da resposta inflamatória desencadeada pela infecção por Toxoplasma gondii, culminando na prevenção de imunopatologia no hospedeiro. Tais achados lhe renderam mais de 1000 citações.
Oi, Ricardo, tudo bem? Parabéns por mais esse marco na sua carreira. Uma trajetória incrível, 30 anos de um laboratório de excelência, onde quem ganha realmente é a comunidade científica nacional e internacional, a população brasileira, certamente. E gostaria de dizer que eu tenho um enorme orgulho de ter feito parte da história do seu laboratório. Foram muitos ensinamentos, muitos aprendizados, muitas experiências enriquecedoras. E espero que você continue sendo esse cientista insaciável, incansável e inspirando gerações futuras. Um grande abraço, espero te ver em breve. Até mais.
Parabéns, Ricardo, pelos seus 30 anos de carreira, liderando os laboratórios da UFMG, da Fiocruz, da UMASS e agora à frente do CTVacinas. 30 anos de contribuições importantes, tanto para a ciência brasileira quanto mundial, e formando pesquisadores que hoje lideram equipes, tanto no Brasil quanto no mundo afora. E um agradecimento especial pela importância que você teve durante a minha trajetória, me orientando desde a iniciação científica até o doutorado, e mesmo posteriormente me dando conselhos sobre decisões importantes que tive que tomar durante minha carreira. Se hoje estou onde estou, você pode ter certeza que você é uma parte muito importante disso. Infelizmente não pude estar aí presencialmente, mas se sinta abraçado e continuo lhe desejando tudo de melhor e mais sucesso, se é que isso seja possível, né? Um grande abraço.
Oi, doutor Ricardo, estou passando aqui para te desejar Parabéns pelos 30 anos de dedicação à pesquisa. Sua jornada é um exemplo de perseverança e paixão pelo conhecimento e que essa celebração traga orgulho e inspiração para muitos anos de descobertas e conquistas. Então, para mim é um prazer trabalhar com o senhor, fazer parte da equipe, porque o senhor realmente é uma força da natureza. Então, mais uma vez, parabéns, felicidades e muitos anos de pesquisa aí pela frente, tá bom? Um abraço.
Olá, meu amigo. E lá se vão quase 30 anos de parceria, de muito trabalho. Muitos devem ter ressaltado aqui antes de mim as suas qualidades como pesquisador, sua inteligência, seu brilhantismo. Mas o que eu quero destacar aqui hoje é a sua garra, a sua energia, força que você coloca em tudo aquilo que você faz para vencer os desafios de se fazer ciência nesse país. Mas é isso que a vida pede da gente. A vida esquenta, esfria, como diz o poeta, se cega, desassossega. O que ela quer da gente é coragem. E afinal, também, tudo vale a pena quando a alma não é pequena.
Com energia incansável e paixão genuína pela ciência, o professor Gazinelli tornou-se um dos grandes protagonistas na luta contra doenças parasitárias negligenciadas. Sua pesquisa tem um foco claro e nobre: o desenvolvimento de vacinas contra enfermidades como a leishmaniose, a doença de Chagas e a malária. Doenças que atingem milhões e, por muito tempo, foram ignoradas. Seu trabalho rompe barreiras entre ciência e sociedade. Não é apenas uma contribuição acadêmica, é um gesto concreto de compromisso com a saúde pública e com a dignidade humana. Desde 2001, seu impacto internacional é reconhecido pela comunidade científica. O professor Gazinelli figura entre os imunologistas mais citados do mundo, segundo a ISI, Web of Knowledge, um indicador claro de sua relevância, originalidade e pioneirismo. Sua trajetória é marcada por feitos que impressionam. Em 1996, tornou-se livre docente pela USP. Na UFMG, foi um dos professores mais jovens a alcançar a posição de professor titular. No ano de 2000, duas conquistas notáveis se somaram à sua jornada. Foi promovido a pesquisador 1A do CNPq e eleito membro da Academia Brasileira de Ciências, em ambos os casos, figurando entre os mais jovens cientistas a alcançar tais distinções, que estão entre os maiores reconhecimentos da ciência nacional. O reconhecimento internacional veio com ainda mais força em 2009, com o Prêmio de Ciências Médicas da Academia Mundial de Ciências, por sua pesquisa sobre os mecanismos celulares e moleculares da resistência e patogênese em infecções por protozoários. Em 2014, recebeu o Prêmio Peter Murani e em 2021, o Prêmio Bung, ambos em reconhecimento ao seu trabalho transformador no desenvolvimento de vacinas e prevenção de doenças infecciosas.
O que eu mais admiro no Ricardo é a sua capacidade de reunir competências em torno de objetivos comuns. Ele tem o talento de fazer a ciência acontecer em grupo, envolvendo tanto jovens pesquisadores quanto pesquisadores sêniores. O INCT e o CTVacinas são a prova disso. Há 10 anos, eu jamais imaginaria que chegaria tão próximo de testar uma vacina contra a malária no homem. Caso isso se concretize, isso se deve à capacidade do Ricardo de impulsionar projetos com potencial de inovação.
E eu acho que ter o Ricardo como um pesquisador no país é extremamente importante, né? Eu acho que tem uma paixão pela ciência, pela formação dos alunos, pelo rigor da ciência, pela qualidade de pesquisa de alto nível, com rigor, né? Que tomar cuidado com os controles, com todos os experimentos. Eu acho que é super importante e, de certa forma, pesquisadores assim acabam influenciando muito as novas gerações, né? Então eu fui um exemplo, né? Fui muito influenciado pelo Ricardo.
Com relação ao brilhantismo científico do Ricardo, eu acho que não preciso mencionar, está aí concretizado nos seus artigos de excelência, nas suas descobertas seminais e, mais recentemente, nessa maravilha que é o Centro Nacional de Vacinas. O que eu queria acrescentar aqui é que a minha admiração pelo Ricardo só cresce quanto mais eu convivo com ele. E realmente a nossa convivência está cada vez mais próxima. Nós fizemos parte da mesma diretoria da Sociedade Brasileira de Imunologia durante um período muito crítico que foi a pandemia. E agora somos membros do Comitê de Assessoramento do CNPq. E é impressionante a responsabilidade e honestidade do Ricardo em cada uma dessas atividades ou cargos que ele assume. Além disso, eu tenho o maior carinho, sou madrinha de casamento e tenho assim, muito orgulho de ter sido madrinha de casamento dele, da Isabela. E digo aqui, pessoalmente, né, ou pelo menos por esse vídeo, que, Ricardo, eu te perdoo por você insistentemente me chamar de Karine. Eu sei que isso faz parte do seu carinho. É isso, beijão. Parabéns por esses 30 anos de sucesso.
E eu posso dizer que se hoje eu me tornei uma pesquisadora, foi graças a todos esses ensinamentos do professor Ricardo.
A ciência brasileira é profundamente grata a você.
Seu nome ecoa também nos corredores de importantes instituições acadêmicas internacionais. Atuou como professor visitante na Faculdade de Medicina de Ribeirão Preto da USP e na Escola de Medicina da Universidade de Massachusetts. Entre 2013 e 2014, ocupou com distinção a cátedra CAPS Harvard, no Centro David Rockefeller para Estudos Latino-Americanos, um símbolo de sua influência científica além das fronteiras do país. Mas talvez o seu maior legado seja humano. Ao longo das últimas 3 décadas, o professor Ricardo orientou com generosidade e rigor dezenas de estudantes, desde bolsistas de iniciação científica a pós-doutorandos, formando uma geração de pesquisadores que hoje lideram seus próprios grupos e projetos, perpetuando o espírito crítico, a paixão e o compromisso que dele herdaram.
Eu queria falar também um pouquinho não só do professor Ricardo como pesquisador e cientista, mas também como pessoa. Ele é uma pessoa extremamente humana, compreensiva, e uma das características que eu mais admiro nele e que eu tento levar para a minha vida é que, assim, se tiver que brigar, ele vai brigar, mas 5 minutos depois ele vai conversar com você como se nada tivesse acontecido. Então, professor Ricardo, eu espero que a gente possa continuar trabalhando juntos por muitos e muitos anos.
Tem sido um grande amigo ao longo de vários anos. Ele foi meu padrinho de casamento, tem me ajudado em várias fases da minha carreira e também me aconselhado ao longo desses anos.
E quero dizer também que, mais uma vez, quero te dizer que eu sou muito grato pela forma como você me acolheu no teu grupo, pela forma generosa com que você lida comigo, sem nunca me pedir nada em troca. Você é, sem dúvida, uma das pessoas mais generosas que eu conheci ao longo da minha trajetória profissional e, por isso, eu te tenho como meu amigo, num lugar especial do coração. Um forte abraço, meu querido, e parabéns. Que os próximos 30 anos sejam tão maravilhosos quanto esses primeiros.
Oi, pai, uma alegria participar dessa homenagem. Meu testemunho vai ser um pouco diferente. Eu vou ler um trecho do meu romance sobre a leveza das asas da noite, em que a imunologia acabou entrando. A malária era uma velha conhecida, desde pequena, da época que papai me levava para o laboratório com ele nos fins de semana. Os camundongos, alguns, tinham malária, infectados por Plasmodium vivax. Numa dessas idas, papai me mostrou como era o P. vivax numa lâmina de microscópio. Levantou-me no ar, fechei o olho direito e espiei com o esquerdo dentro da lente ocular. Vi-os, pequenos pontos rosados espalhados nos glóbulos vermelhos. Não recordo bem que mês era, de que ano ou se estava aborrecido ou feliz. Depois, deixamos o laboratório e fomos até o biotério, onde ficavam os camundongos nos experimentos. As lâmpadas fluorescentes tremeluziam, despejando uma claridade deslavada, lunar. O zunir das lâmpadas e o rumor das geladeiras, repletas de antígenos, soluções, reagentes, imprimiam ao silêncio do Instituto vazio uma paisagem sonora de tédio, interrompida pelos estalos intermitentes das máquinas. Como eram belos os filhotinhos! Não tinham pelo ainda e fervilhavam numa caixa transparente, todos um delicado cor-de-rosa. Menina, tudo o que queria era um camundongo de estimação. Obrigada, pai, e viva a imunologia!
Muita festa, eu sou só papai. Quando eu tava no CTVacinas, eu não fui papai. Pai, você não é vida. Te amo! Quando eu esquecer, eu sou tão papai.
Te amo, papai!
Ele também atuou como conselheiro de importantes revistas científicas e agências de fomento nacionais e internacionais, sempre com o olhar atento de quem busca excelência, mas também com a sensibilidade de quem entende que ciência se faz em rede, com ética e cooperação. Sua dedicação à ciência brasileira foi reconhecida com títulos como de Comendador, 2007, e Gran Cruz, em 2010, na Ordem Nacional do Mérito Científico. E a sua visão vai além da academia. O professor Gazinelli é coordenador do Centro de Tecnologia de Vacinas, CTVacinas. É também fundador da startup Detecta Biotecnologia, que aproxima o conhecimento científico de soluções práticas para os problemas da sociedade. Em 2020, no momento mais crítico da pandemia Covid-19, liderou o desenvolvimento da vacina Spintec, uma iniciativa pioneira que já alcançou marcos importantes, como a conclusão das fases 1 e 2 nos ensaios clínicos da vacina contra a Covid-19. Atualmente, está em processo de submissão à Anvisa para aprovação a proposta de ensaio clínico de fase 3 da Spintec. Em 2022, deu mais um passo histórico com o início das obras do Centro Nacional de Vacinas, um sonho antigo que começou a se materializar, oferecendo ao país soberania científica e tecnológica no desenvolvimento de vacinas.
Eu também queria agradecer por ter sido meu mentor, meu orientador no meu doutorado e a pessoa que abriu as portas da imunologia na minha vida. Muito obrigada por tudo que você fez, por me ensinar a fazer uma pesquisa de ponta, uma pesquisa internacional. Obrigada por ter aberto o mundo da imunologia, né? Aberto portas internacionais para mim. E hoje eu só sou quem eu sou como imunologista porque eu tive você como meu grande precursor. Muito obrigada e por tudo que você faz por mim e por todos os novos pesquisadores na imunologia. Parabéns.
Ricardo é sempre muito criativo, é muito alegre, um pouco ranzinza, como todos Gazinelli, mas ele também é um grande amigo e compartilhar com ele sempre foi um grande privilégio. Nunca fomos concorrentes, sempre fomos colaboradores e continuaremos assim. Ricardo, parabéns por tudo que você fez e muito legal essa sua comemoração. Continue com todo esse sucesso e essa vontade de fazer. Um forte abraço.
Eu fico muito feliz, muito orgulhosa de fazer parte desse grupo e de conseguir chegar onde a gente está chegando sob a liderança do Ricardo.
Eu fico muito emocionada de fazer parte dessa história do laboratório. São 30 anos de pesquisa e eu fui uma das primeiras alunas dele. Então, é muita responsabilidade estar aqui e a única coisa que eu quero falar mesmo é eu agradecer ao Ricardo por esses maravilhosos anos de convivência e agradecer a todos, né, que fizeram parte dessa história. Um abraço, um beijo.
Fico muito contente por ver essa trajetória até aqui, né, e fazer parte dessa homenagem tão bonita para o Ricardo. Acho que todo mundo de parabéns, o grupo, o Ricardo e todas as pessoas aí que ele formou. Eu acho que realmente é merecidíssima essa homenagem. Eu acho que é um processo super importante aí e eu fico muito contente de fazer parte desse grupo e fazer parte dessa homenagem. Então, parabéns, Ricardo. excelente de trabalho, precisamos de mais pesquisadores como você no Brasil.
Então, o que eu posso te desejar, meu amigo? Muita força, muita energia, muita intensidade para continuar enfrentando os desafios, porque o futuro está logo ali, o nosso CTVacinas.
Professor Ricardo, sua vida é um testemunho vivo do poder transformador da ciência, guiada pela paixão, pela ética e pela generosidade. Sua visão estratégica, sua curiosidade incansável e seu compromisso com a saúde e a educação pública são fontes inesgotáveis de inspiração para todos nós que tivemos e temos o privilégio de caminhar ao seu lado. Neste dia tão simbólico, em que celebramos 30 anos de uma carreira científica marcada por conquistas extraordinárias e dedicação inabalável, expressamos nossa mais profunda gratidão. Gratidão pelo cientista brilhante, pelo mentor generoso, pelo líder inspirador e, acima de tudo, pelo ser humano extraordinário que o senhor é. Muito obrigada, professor Ricardo Tostes Gazinelli, por dedicar sua vida à ciência, à saúde e à humanidade. Seu legado é imenso e seguirá vivo em cada pessoa, cada ideia e a cada vida tocada pelo seu trabalho.
Meus parabéns pela sua carreira, pelos netos lindos que você nos deu. E seu pai e eu sempre fomos muito orgulhosos de você. Um beijo.
Parabéns por essa história linda na ciência. Você é um exemplo para todos nós. Eu tenho muito orgulho de você.
Nós te amamos.
Nós te amamos.
Eu não vou falar muito, né, que eu estou emocionado, mas as duas últimas pessoas, eu tenho uma história boa com as duas últimas pessoas que apareceram, que foi minha mãe e a Isabela. Então, nós estávamos em Boston e fomos jantar no Legal Seafood. E aí eu fui para o banheiro e a Isabela começou a me elogiar, né? Com nice I was. Depois eu vou traduzir para o Alan. Quão bacana eu era, quão gentil, quão inteligente e tal. E passou um pouco, minha mãe falou, será que nós estamos falando da mesma pessoa? I'll translate to Alan. Let me translate to him. Let me translate to him. Okay. So, anyway, eu tenho muito que agradecer. Eu acho que quando eles falam de sucesso, esse sucesso realmente é do pessoal que trabalhou comigo. Eu não tinha na bancada costumava dizer que eu tinha duas mãos esquerdas, né, que não era muito bom, e com certeza eu acho que foi a dedicação de todos vocês que estiveram comigo que fez. E a motivação, ela também veio de vocês, né? Que eu via, cada vez que eu via um gráfico, eu ficava tão animado. Então, obrigado a todos aí. Obviamente, eu tenho que agradecer a Isabela, aí, que tem sido aí um grande apoio para mim, e também as minhas meninas, né, e meus meninos, que também são uma parte muito importante na minha vida e que me dão muita alegria. Minha mãe também, meu pai, meus irmãos. Aí, se eu for pôr todo mundo, eu não vou parar de falar aqui, mas realmente tem sido... Não tenho... Já reclamei muito da vida, né, Alan? I did complain a lot about life, but in general, I don't think I have to complain, and I think that I was always blessed, yeah. And I'm glad to be here, and I'm glad to be here with all of you.
So, hello everybody. Well, I was told that now we will turn to English. That's what I was told. Actually, I thought that after this incredible ceremony, we would go to a bar, drink, and then spend the afternoon just talking, chatting. But I will be even briefer than Ricardo. For the sake of time, it's just a great pleasure for me to be here to chair this incredible session with 4 amazing speakers who had contributed a lot to science, technology, innovation. So I'm not going to spend time showing their CV here. So I suggest you guys to go to the internet and check all the awards and publications. So I'll be really, really brief. And the first speaker, I don't see, Dr. Andrew Simpson. All right, there he is. So it's a great pleasure to introduce the first speaker, Dr. Andrew Simpson. I'm just going to mention briefly that he was the coordinator of the first complete genome sequencing in the South Hemisphere, the Shilela project, and thereby contributing a lot to the molecular biology in our country. He was the former scientific director of the Ludwig Institute for Cancer Research, now the CEO and for the Orygen Biotechnology. Okay, thank you very much, Andrew. You speak here.
Hello, everybody. Hello, Alan. We haven't met for, I think, 40 years. You're looking much better than I am. Congratulations, Ricardo, and thanks very much for inviting me to give a talk. I'm going to talk about my experience in Brazil. I feel a bit embarrassed because it's all about you, really. So forgive me. Okay. Melhor? Okay, that's me. So Ricardo asked me to give a talk about my experience and my opinion about being a researcher in Brazil, right? Good. At least we're on. Thank you very much. No better example than you. But anyway, I started working in Brazil in 1990, quite a long time ago now. And prior to that, I'd been at the National Institute for Medical Research in Mill Hill and NIH with Alan. Two of the best places in the world to do science. And I came here for lots of reasons, but mostly because of Brazil, because Brazil is a wonderful country. And I also discovered soon after I got here that if you're a scientist, if you're born a scientist, you're always a scientist. You're always going to do science. There's no choice. It's part of your blood. And so I was here, and I thought, what are we going to do? And I realized two things. I've had two rules about doing science in Brazil. First of all, Brazilian scientists are the best anywhere. I'm not saying they're better than anybody else, but I've never been anywhere there's better scientists than in Brazil. So what's the best thing you can do in Brazil? Collaborate. Because if you collaborate, you know you're going to be working with somebody really smart. And I'm not just saying that to be nice. It makes sense. It's very hard to become a scientist in Brazil. There's a funnel. There's a lot of people born. Few people get to university. Very few people get to have their own lab. So this is a funnel of brilliance. And you go anywhere in the world, and I think Alan's lab is an example, you'll always find a Brazilian scientist there. Any lab in the world. And they're always one of the best. So that's the first thing, collaborate. The second thing is, what's the big problem of doing research in Brazil? Importing reagents. It's as simple as that. It takes forever. It was like that when I came, and it's still like that. And what does that mean? That means if you have a really smart idea today, probably somebody else somewhere in the world is going to have it today too. Because we all stand on the shoulders of giants. None of us have ideas that come from nothing. They come from reading the literature. They come from the work of others. You have a brilliant idea, and you need a reagent for it. But the chap in New York who had the same idea will have his reagent tomorrow, and will be publishing his paper before your reagent arrived. So those are the two things about doing science in Brazil. Collaborate and think ahead. You have to know what reagents you want 6 months from now, because it's the only way you can compete. And basically that's what I did. And I'm just going to tell you a story about my life in Brazil, where basically I only used one reagent, DNA polymerase. And I used it for doing PCR and sequencing. And that's all I did. And let's start here. So when I first arrived at Unirashu, there was a PCR machine. And PCR had pretty much just been invented, and people were playing with them, and a few groups had primers for doing PCR. And there was a group there who had primers for amplifying, I think it was the ribosomal DNA gene, the ribosomal RNA gene of Biomphallaria glabrata. And if you do a PCR of different species, what do you get? A single band of that gene. But playing around, what we realized, if you lower the temperature just for 2 cycles to a lower stringency, those same primers will bind to lots of places in the genome. Then you go higher the stringency back up to what it was meant to be, and you just do a normal PCR. And what do you generate? You generate a whole pattern of bands that look like this. But it's not just a mess. They're all specific interactions, but of lower stringency. And they make up a DNA fingerprint. And if you take away the specific band here, which you can hardly see, well, you can realize if you took those primers and did the same low stringency PCR with any DNA, you're going to get a pattern. And that pattern's going to tell you something. And that was the first thing we did. And that's basically all I ever did at Unirashu, is low stringency PCR. Because we had a few primers and lots of bright scientists with all kinds of phylogenetic questions and problems. And we used the same primers and did it over and over again and published loads of papers. And this was just one, where here you've actually got different species. And here you can see 2 different species simply because of these low stringency bands. And it turns out you can start doing all kinds of interesting things with this low stringency PCR. And here, for example, is one talking about collaboration with Sergio Pena, great scientist from here, Fernando Heineck, Emmanuel Diaz Neto, who really was my great partner in all this science. Here we showed you can take a single gene and do low stringency PCR and pick up point mutations. And here, this was Sergio's work, you can show this was mitochondrial DNA with the D-loop primers, and you can lower the stringency and see mother, child, father triplets. You can see this is mitochondrial DNA, so the mother and child are identical, and those patterns are absolutely identical, and the same here. And this is a paper I did with Otávia Cabeleiro and Luisa Villa in São Paulo, which showed that you can also quantify by using these low stringency bands as an internal standard. And here we were quantifying the amount of papillomavirus in samples. And this was so good and so accurate that this technique was actually used for the clinical trials of the HPV vaccine that Luisa did in São Paulo. So this work all went very well here in Belo Horizonte, and actually I got offered a job in São Paulo, and I went. And when I got there, soon after I got there was an announcement made that the scientific community in São Paulo is going to sequence, it says, going to sequence the first plant pathogen. Well, I thought, well, that's great. I'll join in. I did join in. But what I realized, this was a terrible thing to do. Put an announcement in Nature of what you're going to do before you've even done it. Talk about putting on a little bit of pressure. Anyway, we live with that. And this was really a superb example in my mind of what I'm trying to say about science in Brazil. We take this list of people. These were the heads of the labs who joined in with the sequencing. All fantastic scientists. And this is collaboration par excellence. And it's also following my mantra. We know what we're going to be doing this year, next year, and the year after. Sequencing DNA. What do you need? Sequencing kits and sequencers. That's it. And all of the intellectual part was done on the internet. We actually had to install telephone lines at a lot of people's labs, including mine, to join the internet. But it just shows that if you take away the limitations that we have in Brazil that are no fault of our own, Brazil competes at the very highest level and does as well as anyone. There's no customs on the internet. So the information goes backwards and forwards, and we compete with anybody. And in the end, having announced this, nobody wanted to coordinate the project because it was obvious, what could go wrong? It's a really good idea for Fernando Perez and Fernando Heineck. It had all the funding you could want. It's only going to go wrong if the coordinator's rubbish. So nobody wanted to do it. So I said I'd do it. João Setúbal, João Meidanes did the part of the bioinformatics, and we had a steering committee from Europe, which was great because they were meant to tell us how to sequence genomes. The trouble is none of them had ever done it, and the information they gave us was completely wrong. However, we did sequence that genome under budget, ahead of time, and because of the really smart people involved, they were able to draw all kinds of inferences about the life cycle, the life of this pathogen. We had lots of sessions like this. This is at the Ludwig Institute in São Paulo. Probably people you recognize, all just sitting around looking at the sequence, all coming up with ideas about what we can write in the paper. And we were lucky enough, it was published in Nature as an article, was on the cover. The leading article, when you open it, was about the project. And really, I think it was just an example. If you have a level playing field, you can do anything here, because you've got really smart people. And Brazil celebrated this in a way for which I'm very grateful, because it was lots of fun on the newspaper, in the magazines. There's Mario Covas, who was the governor of São Paulo at the time. The president, Fernando Henrique, we went lucky enough to go to the palace. even got internationally known. But this was the thing that I really treasured the most in The Economist, which had this article started, Samba, Football, and Genomics. The list of things for which Brazil is renowned has suddenly got longer. I think we should say science. Brazil has become a great country of science. This is one of the things I enjoyed the most was even American newspapers interviewed us and wrote articles, and this was one from the Los Angeles Times. And it was about scientists here. And Pierce's disease is a disease of Shigella, of vines, which is a big problem in California. And what was really nice, a school teacher saw this and got all the children in her class to write us a letter in Brazil thanking us for sequencing the Shigella genome. And I think it's my biggest contribution to humanity that we did help save Californian wine. Even from here. So, this was all well and good, but I had gone to São Paulo, and I was working for the Ludwig Institute for Cancer Research. And we had a visit from the board of directors. And they said, we've heard you've got this chap, Simpson, he's doing a very good job, but he's working on an orange pathogen. What's that got to do with cancer? They said, it was orange juice because it's good for cancer or what? And he said, and our director, Ricardo Brentani, said, well, we're about to do a project on cancer. He hadn't told me, but he told them. They said, that's all right then. And this then was based on work, the same work that we've been doing here in Reni-Rashu, where instead of taking DNA and amplifying under low stringency with randomly selected primers, you can amplify RNA. And if you do that, you amplify, obviously, expressed genes. And it's a way of generating sequenceable bits of genes, expressed genes, ESTs we called them, messenger RNAs. And that was very, very important at the time, because this was the time when the human genome sequence was going on. If you sequence the genome of a bacteria, you can identify the genes very easily because they're contiguous. You get one, you got a little gap, you got another one, you got a little gap. The human genome is 97% non-coding. And if you just sequence the DNA, you can't really see the genes. So it was critical that somebody sequenced the expressed genes so you could map it against the DNA, human DNA, and actually see where the genes were. And we then invented this technique, which we called, I can't remember what we called it, but basically we were generating bits of expressed genes and sequencing them. At the time, what everyone else was doing to see an expressed gene was to do it from the 3' end, an oligo-DT primer, and you sequence. And there's two problems with that. One, you go into the 3' untranslated region, and you can't really see what the gene is. Secondly, you're going to amplify based on the proportion of the different RNAs that are there. So common genes you're going to sequence over and over and over and over again, and the rare ones you never sequence. This is based on sequence, not on abundance. So you get a much better sampling of the population. So the Ludwig Institute and FAPESP both put up a good amount of money for us to do this with RNA derived from tumors, which is basically the human genome. And again, it was announced in Nature, but this just shows an example from one of our first papers, which shows if you take the RNA that we amplify randomly and just show where it goes through known genes, you can see we sequence from the middle of an RNA, whereas the 5' and 3' ends, obviously you sequence the end. So those three pieces of information together actually is this that actually identified the genes in the human genome. And we teamed up with the NIH and submitted all of the information together, and it was all used in the Human Genome Project. This is just showing how important the information is. This is a human DNA. This is coding. The red is coding. The black is non-coding. It's really impossible to see a gene unless somebody tells you that it's there. So skipping over all of that, the human genome was published in 2001, and this is something I'm very proud of. In this, in the initial article, it says there appear to be 30,000 to 40,000 protein coding genes in the human genome. That's wrong. If you look at what we did and take all of our data, we estimated at the same time there are 23,000 genes in the human genome. And you look in Wikipedia today, how many genes are there? 20 to 25,000 genes. So we actually got the genes right. So there, Craig Venter. And so the last project I'm just going to talk about actually went on concurrently, but you can see the theme here. We're always collaborating, we're always doing the same things over and over again so we know what we want to buy. And CMPQ are nice enough to ask me to organize a project that involved labs from all over the country. And Santuza's lab here in Belo Horizonte was one of them. And we sequenced the genome of Chromobacterium violaceum, which is a free-living bacterium found pretty much anywhere, but we really wanted to say it was from the Amazon. These are all the collaborators. This is one of our sort of getting together days at the end, working out what all the genes were and everything. And this is where everything just comes a little bit full circle, that when you sequence a genome, and this is what we were told to do by the gentleman on our steering committee, the way we did it was to make cosmids of about 10, 12 kb long. We divided those out between the different laboratories. Each laboratory sequenced the cosmid. Then you put the sequences together. The trouble is, there are always gaps. There are always gaps between your cosmid contigs. In the Shillelagh project, we did everything we could think of to join out those gaps. Basically, we did outward-facing PCR, we did hybridizations, and we couldn't find a way to do it. And the steering committee actually didn't know what to do either. And though this isn't widely known, what we actually did was resequence the genome the way it should be done with small plasmid-sized fragments. And we quickly closed the gaps because they were actually quite small. And we had a lot of information, and 3 months after we started resequencing, we had the genome again. The trouble is that was very expensive. In the CMPK project, we didn't have any money left. So I thought, what are we going to do to close the gaps? And we invented a technique, and it was the same thing that we did before, low stringency PCR. So basically what we did is, well, let's just go back to this, it's easier. Let's take this gap. What you do is you make a PCR primer for that end, and you do one round of high stringency PCR, and you're going to extend into this gap. You don't know what the sequence is, but you're going to extend into it. Then you do a low stringency round, and that primer will bind somewhere. Primers always bind somewhere. They'll bind the best match, and then come back, and then higher the stringency, and go backwards and forwards in a normal PCR. And what you do is generate a fragment into that unknown end. We just did that for all the ends, and it worked very, very quickly. In fact, it was amazing. And we sequenced the genome, we published it, and I got a new job in New York. There's two things I learned after going to New York. I had a great time there, a great job. I became the director of the institute. But two things I learned. You can't direct science. It doesn't mean anything to be a scientific director. You can't say to someone, go and discover that. All you can do is basically evaluate people's work, decide funding, and so on. The second thing I discovered is there's no better place in the world to live than Brazil. And I was always waiting for somebody to make me an offer I couldn't refuse to come back. And one day they did. And on Friday, I'm going to give another talk and tell you what I did when I came back. But anyway, this is Ricardo's day. Sorry to butt in with my story, but that was my advice on doing science in Brazil. Collaborate and keep it simple. Thanks a lot.
Thank you very much, Dr. Simpson. Are we going to open for questions? Are you guys, or should we leave the questions as we did in the morning for the end? Help me. Thank you. Okay, thank you very much. So now I don't see either. All right, here she is, my dear friend Lis Antonelli. So Lis has been contributing quite a lot on the field of immunology, infectious disease, also a lot to the Brazilian Society of Immunology. She's the senior, I think most of you know her, so no place for me to introduce, but anyway, so this Lis is senior scientist at the René Rachou in the Fiocruz, and also has a joint position in the University of Massachusetts. Still? Right? More or less? Okay, Lis, thank you, help me. Thank you very much.
So, good afternoon, everybody. Okay. Can you put my presentation, please? Okay, so Ricardo has left. Okay, so I have to wait? No, he's gonna cut me as well. Well, I'm thrilled to be here. Well, we're gonna see that we had a long path walking together, and actually I was a little bit nervous when I started doing this presentation and thinking about the 30th anniversary of Ricardo, because when I started to put together my story, because he asked me to talk about my story as well, that I found a little bit weird, I discovered that I've known him for more than 20 years. So here is a little bit of my story. I'm not going to talk a lot about that, but I'm going to start when I came to Belo Horizonte. So the only STED element that I had in my career was immunology. I moved from place to place and from disease to disease, but immunology stayed as an essence, I have to say. So during my master's and afterwards, Gustavo and Karina can talk with you about my master's. I'm not going to do this. I came to Belo Horizonte twice. So I was helping a friend and a colleague to do experiments with flow cytometry. So in São Paulo, I was doing human mouse malaria. Then I was coming here to process some PBMCs from malaria patients from the Amazon. And then the second visit, Ken invited me to do my PhD here, and I came. And I have to say that if there's a word to summarize what my career is adaptation. So things were not too great in São Paulo, then I decided to come to Belo Horizonte. And I started doing human leishmaniasis, and I was doing several times going several times a year to work with this guy here, Dr. Edgar Carvalho. That was a really pleasure for me. He's a giant scientist. And also doing leishmaniasis, I was to University of Oxford at Fiona's Powery Laboratory. But there, I have another problem. I couldn't ship my samples. So I was like, what am I going to do here? I don't know. And it lasted like 2 days, and I realized I had to make myself available. Then I offered, I don't have my project anymore. Can I work with you? Any project. And then Fiona put me together with Kevin Malloy, And then I started working with gut inflammation and Helicobacter hepaticus here. So it was like a total different story. I went back to mouse model again. But it was great, actually. And we have, actually, the best thing was that Kevin is still my collaborator. So we had even a student, two students together afterwards. And then I went back to Minas Gerais, to UFMG, to here. And this was my first encounter with Ricardo. So when I came back, I had done a lot of CBA, and Ricardo was interested in this technique. So he asked me to do some experiments to finish Bernardo's project. And I said, oh, I have to finish my PhD. I have papers to write, thesis, and I don't have time. But you know Ricardo, he never takes a no for an answer. And he said, no, it will be only 1 or 2 experiments, and you can do it during your weekends. And I can come and work with you. And I said, oh, I was like 20 years earlier, I was like, okay, so I can do the experiments. And so he did. He came to do the experiments with me for the first weekend, and then he had also a technique and an undergrad student. And then we talked, and we said, let's talk to Ricardo to stay at home, that we can handle the experiments. So we did it. We finished the experiments, and this is our first paper here. And a year later, actually, Ricardo was in my thesis committee, and I had 2 Gazinellis in my thesis committee. It was like an adventure. And I also had Leda and Alda, that are giant scientists. So I was very happy, but I lost a lot of weight because it was a stressful time as well. So then I went to NIH, and this was fun, but actually I have to adapt again, because I asked Alan, actually I sent a letter, and I asked for a position. I told him I'm gonna apply for PIL, and I want to do a project with mouse and monocytes and leishmaniasis. And then he answered right away, because of course I had 3 very important reference letters. I had here Ken, I had Fiona, and I had Gazinelli. So it was like 2 days, and he said, yes, you can come, but I don't want another mouse immunologist. You have to come to do human TB. And I said, that's okay, I can do human TB. But then another challenge, because it was hard to find TB at NIH. So in the beginning, I had to sneak in, and I started a mouse project against Alan's will. But to make Alan happy, actually, I contact with people from clinical center. I started taking samples from patients with IL-12 mutations and this kind of things, until I found Irene. Actually, then found Irene, and she wanted to develop a model of IRAS, immune reconstitution inflammatory syndrome. And Irene wanted someone to work with her patients, and Alan wanted to work with TB. So what we did, we established a collaboration with Tuberculosis Research Center in India, and we started this project. And on top of that, I work with Irene in immune reconstitution inflammatory syndrome with patients with HIV. So here I had at least 3 projects. It was a very hectic time, and Gazinelli appeared in my life again. So he called me and he said, you know that Fiocruz are going to open some positions, and you are the first candidate, something like that. And he said, you can come for my lab. And I said, I have a lot of things to do. It's very hard to move now. And he proposed me, no, actually I proposed him. I can go, but you have to let me to go back to NIH for a year and finish my things. And he said, okay, you come, but you promise you have to come back in 1 year. And then I did, right? After a year, I started in Ricardo's lab, Laim. And this time was really great because Ricardo always have a lot of very smart people in his lab. And here I shared with Ricardo the mentorship of 4 PhD students and 2 postdocs. Samantha, she's in the back. So she was working with mouse toxoplasmosis. Then Miriam was doing with Samantha as well a work in the endemic area of tuberculosis. And Luara was doing cancer, right? She was working with this, the vaccine, the T-cruzi expression IS1. And afterwards, actually, Luara became a postdoc in the lab with Kevin, Ricardo, and myself. And then Bruno was a, very sweet guy. And we had a great paper together. It was a lot of work, but the paper was great. And Fabiana was working with malaria, and Fabiana and I worked very close together. And the last one was Bruno Rocha that helped us to finish up a project with human toxoplasmosis as well. So it was a great time. I supervised a lot of students, and I was going very often to the Amazon. So I don't know how many times and how many students I trained there. And we established a very strong collaboration that was initiated before with Bernardo, Gazinelli, Douglas, etc. And then after a while, I became a mother. So actually, I was like huge, going to Porto Velho, working a lot, not sleeping at all, but it was fun. And I had like students having babies as well, so it was great, but was very busy. And when I had my second kid, actually, I said, oh, now I have to stop. I have to slow down, and I have to do as if it was true. I have to have my own lab. So I don't know if it was a good choice, because then I had to work twice, but then we continue our collaboration. Actually, someone told that Ricardo stopped doing malaria. I think it was Julia, but Ricardo continued doing malaria, and we continue working together. So this is my team today, not today, but it's my team that I built from the days of Ricardo's lab to today. And now actually I have 2 PhD students, 2 masters, a postdoc, and Luara stays with me as a visiting researcher. And this is the connections I did during this time. And one thing that I have to say that building scientists is a really team effort. And it's much more important to have collaborations than competitions. So I try to keep this in my mind. And if you collaborate, everything is going to be fine, right? So we have to adapt. And if life gives you this, you have to do whatever you want with this. Or if you like lemon, you take lemon as well. How many minutes do I have? Okay. So, and for the second time, Ricardo also asked me to talk about anything. So, actually, I choose something that Ricardo was really, and still is interested in, that is immunometabolism. In our case, it's immunometabolism in malaria. So we started, we have 4 publications up to now, and I'm going to talk about this first 2 today, very, very briefly. Here. And once upon a time, I came to Brazil, 2009, and I was with a lot of ideas in my mind and a lot of hypotheses, and I like to keep working with what I was doing, but Ricardo said, anything but TB. And then I had to adapt again. And I started doing a little bit of everything, as you saw before. And actually, the focus on the postdoc that I was working with was neutrophils. So doing these experiments, what I noticed was that actually these patients had very strong monocytosis. So I said, oh, I was working with monocytes at NIH, actually with Hiltonol that you talked about a lot here. And I said, oh, let's go for that. Why not? And I'm going to cut this story short. This is part of the story. And we did some electron microscopy only because I really wanted to do microscopy in my life. I love images, but I always do flow cytometry. And everybody says, Lis, flow cytometry. I wanted to do, and I went to do electronic microscopy. So what we notice here is that the monocytes, here we have the 3 subsets. The monocytes from malaria patients had this very morphological characteristics of activation. And when you compare the 3 populations here, and here I'm showing only the Plasmodium vivax monocytes, we see that there's a lot of mitochondria. And if we count the area here, if you measure the area, these 2 subpopulations, classical and inflammatory monocytes, have mitochondria occupying greater area in monocytes. And we also saw that these mitochondria were more active in patients first, and then when you compare the 3 populations in inflammatory monocytes. So we started doing some functional analysis. And as a brief aside here, I have to tell you that for those who do not know biology of Plasmodium vivax, we cannot culture these parasites. So it was really hard to do these experiments. We had to have 100 ml of blood of each patient to purify the infected reticulocytes and do the experiments. So what we saw was that inflammatory monocytes phagocytose more the parasite. They kill better the parasite as well. They produce more TNF, and also they produce more reactive oxygen species. And most important, they produce more mitochondrial ROS. So what do you know here? We know that metabolic changes occur during stress. So we have infection, we have stress. And the host have to adapt, right, to fight against this infection. And what became a paradigm in immunology now is that oxidative phosphorylation metabolism turns to glycolysis here during stress. And then, as we saw that infected monocyte produce a lot of ROS, we try to explore the metabolic profile of these monocytes. And here, we look for glucose uptake, and we see that because glucose is a good source of ATP only during stress. So we wanted to see how was glucose uptake here. And we saw that inflammatory and patrulling monocytes was doing more glucose uptake than the monocytes from healthy donors. And we also induced stress in the mitochondria to see how they behaved. So in here, we measured the oxygen consumption rate, and we saw less basal respiration, less maximal respiration, less proton leak, and ATP production in Plasmodium vivax, in monocytes from Plasmodium vivax when we compare to healthy donors. And to explore this more, what we did was to look the genes that were associated with mitochondrial metabolism. And in here, I'm only showing actually the genes that were different from Plasmodium vivax infected patients and controls, okay? And we see here an overall reduction of several genes involved with fusion and fission, apoptosis, metabolite transport, chaperone activity, protein processing, electron transporting chain. But we saw some very punctual increase here. We saw an increase in SOD2, that is enzyme involved in antioxidant activity, and also this A37 involving iron metabolism, and PKM here, an enzyme that participates in glycolysis. And then if you see here, actually in this PCA, we see that actually monocytes from Plasmodium vivax occupy a very different transcription space compared to healthy donors, but the same is not completely true when we try to separate the monocyte populations here. But actually, we see differences. So if you see healthy donors and monocytes from Plasmodium vivax infected patients, we can see that actually these three subpopulations share a lot of genes when we look for monocytes from healthy donors, but only few genes are shared in monocytes from these Plasmodium vivax patients. So this suggests that these monocytes become a lot more specialized when they see Plasmodium vivax. And then here, the problem is it's not easy to control parasites. Actually, it can be easy to control parasites, but we have to control the immune response as well. So we need a balance here. And one way to balance is to produce the inflammatory mediators when we need them and where we need them. So what we did here was to do imaging using image flow cytometry, and we saw that, like, we purified, again, monocytes, and we did some colocalization assay, and we can see here that these monocytes phagocytose the parasite here, and this happened more intensely in patients than in healthy controls. We saw also that actually the recruitment of mitochondria doesn't change to the phagolysosome doesn't change if we look the Plasmodium vivax or healthy donors. And however, we see more colocalization of Plasmodium vivax and phagolysosome. And here we see also colocalization of Plasmodium vivax and mitotracker, the mitochondria, and also the Plasmodium, the parasite, and the production of mitochondrial ROS here. So what we see here that, at least in our experiments, we see things happening very colocalized. So when you have phagocytosis here, we have recruitment of mitochondria, and we have mitochondrial ROS being produced. So this is actually this is Sueli. She was with me since her undergrad training, so he followed all since the first paper. And here we did this summary so you can understand what I just told you. And during the steady state, what happened with monocyte? Monocyte produce a lot of ATP via OXPHOX. So consume this oxygen, and we don't have a lot of reactive oxygen species, and we don't have a lot of disturbed mitochondria. So the membrane potential is low. However, when we have phagocytosis of Plasmodium vivax infected reticulocytes, there's a metabolism shift. And what happened here is that glucose start to be uptaken for these monocytes and actually mitochondria colocalize with the phagolysosome. There is a decrease in oxygen consumption and a decrease also of ATP produced by OXPHOX. And what happened with the mitochondria is that there's an increase in membrane potential. So we have actually a break, and complex 4 is not working properly anymore, and this makes the production of mitochondrial ROS increase. And then we have the parasite control. So I'd like to thank Ricardo. I think it is a pleasure to work with him, and I'm thankful for a lot of help that he has given me during my career here. And as someone also already said, I'm not sure if there's a way to have more success, but I congratulate you, and thank you.
Thank you very much, Lis, for your beautiful, wonderful story and great results. Again, we're not going to be open for questions. And with that, I will introduce the next speaker, Doug Golenbock, who came before to me and said, no, don't say anything about me because I will be introducing myself during the presentation. But I want to acknowledge anyway that he's professor of the Massachusetts University Chan Medical School, and he also holds the Neil and Marjorie Blacklow Chair of Immunology and Infectious Disease, and has been a really huge contributor to innate immunity. I need to mention the tall meetings that he initialized a long time ago. Everybody works with innate immunity, and attended to one of these meetings is just unforgettable. Okay, thank you very much, Doug.
Well, it's really great to be here on this day. 30 years, huh, Ricardo? You don't look that old. Anyway, I have to apologize to you. I had a short version of this talk, and I brought it up to the room upstairs, and it was corrupted. So I'll be skipping a bunch of slides because I have to use the longer version of it. So, all right, so this is about malaria, and it's about Brazil. Ricardo said, tell the audience about Brazil and you and me and whatever. So I thought I would actually start with a little bit about myself. That's me in 1954. Which is the year I was born in. This is something Ricardo and I have in common, actually. I was born more or less about there, okay, and I think Ricardo was born somewhere about there. Right. So you're on the Upper West Side, and I'm on the Upper East Side, and there's supposed to be some type of competition between the Upper West Side and the Upper East Side, but I'm not sure what it is. Anyway, we have that in common. And after I was born, I almost went immediately to medical school. I went to the University of Michigan Medical School. I actually went to college in Michigan, too. And then I went to George Washington University to do a residency. And then I was, decided to become an infectious disease specialist. So I did my infectious disease subspecialty training. And I finished it all, and I took my boards, and I was board certified in infectious diseases and everything. And then I met this guy here, who he's now dead. He was a biochemist, and he was one of the first lipid biochemists in the world to use genetics. Actually, he may have been the first lipid biochemist in the world to use bacterial genetics, and he developed all these techniques for making mutants and analyzing them biochemically, and he just completely corrupted me, and I no longer wanted to be a doctor. I wanted to become a scientist, and this was much to the chagrin of my parents, who had put me through medical school and all this stuff, and I was already trained, and I was supposed to be out there actually making money, but instead I was working like a graduate student in a biochemistry laboratory at the age of I was about 50 by then. So I was pretty old, but I got to see some great scientists. I worked at University of Wisconsin. I did a little stint at the Rockefeller and then MERK research labs, and it was great. And I came up with this idea that I really wanted to find a big scientific question to study, and I thought, well, the good one would be, since my boss worked on E. coli lipids, and he in particular, he was interested in LPS, I thought, well, maybe I'll just figure out what causes sepsis. Sepsis is a pretty big question. There are millions of cases diagnosed annually, and it causes over 50% of deaths in the hospital. So that's a big question. And of course, it's for most people think they understand what sepsis is, but I can assure you that there is no good definition of sepsis. People come around to this every 2 years. They have a meeting where they try to define sepsis, and no one's come up with a very good definition yet. But we all know what it is when we see it, as Justice Stewart once said. I think only the Americans in the room understand that one. But in any case, people get low blood pressure in their heart, and all their organs start to fail, and 20% of the time they will pass away from multi-organ failure. So this is a typical gram-negative outer membrane. This was actually from a review article written by Chris Rates just before he died, and I still use it, even though it's a little outdated. The whole point is that gram-negative bacteria are coated in LPS. So it seemed to everybody that LPS was the cause of gram-negative sepsis. If I can get through this, yeah. So, and the reason we say that is because you could give it to humans, and it looks like they've got sepsis, and there are actually studies that have been done where they give LPS to humans, believe it or not. And for many years, it was thought that LPS was the cause of gram-negative sepsis. And when I'm telling you, this was like dogma, that sepsis was caused by LPS. In fact, I even knew people who said that gram-positive sepsis was caused by LPS. That's how deep the dogma was about the whole thing. So the big question is, what's the receptor on cells that recognize LPS? And this was really a big question, and there were many groups that were racing to find the answer to this. And I will say, from having been there at the time, that it was really a tie between these three guys right here, okay? But the first one to have a paper in press was the guy at the front here on that beautiful horse there, Bruce Beutler. I think you all recognize him from the way he is. And there he is a little bit later on when he collected his Nobel Prize for the discovery that toll-like receptor 4 was the signaling receptor for LPS. Doesn't necessarily mean it's the cause of sepsis, though. That's a whole other question. It turns out that there's another part to this receptor called MD2 that was discovered by Kensuke Miyake in Japan, and it's just as necessary as toll-4 is to recognize LPS in cells. Toll-4 doesn't work without MD2, but somehow the Nobel Committee missed that one. But there aren't enough Nobel Prizes for all the people really who deserve them. But that left me feeling kind of blue about this great big question. So I needed another big question. And that's when I was at this Keystone meeting and I ran into Ricardo, who we were talking, and he convinced me that we should study malaria together. And I thought, that sounds like a great idea. Do they have malaria in Brazil? I really didn't know if they had malaria in Brazil. And he said, yeah, we have malaria in Brazil. Turns out there's not all that much malaria in Brazil, but there's malaria in Brazil. And it's an endemic region, especially in the north. And we started working on this together. So this is a beautiful cartoon of the life cycle of a malaria parasite. And the part of the life cycle that interested me the most was this thing called the paroxysm. And that's when erythrocytes burst and they release all their inner contents into the blood, and people get shaking chills and high fever, and sometimes a lot worse, multi-organ failure and death. And it's actually kind of like sepsis, okay? And some people like to call it malaria sepsis. And so the paroxysm of malaria is this inflammatory cytokine storm. People have measured high levels of cytokines during paroxysms. Temperature can be very, very high, like 40 degrees centigrade, and the consequences can be lethal. And there are, indeed, hundreds of thousands of people every year who will succumb to malarial infection. The epidemiology is a little bit soft on malaria, but we know that there are hundreds of thousands of people who die, and there are millions of people who get the disease. So I thought this was going to be my quick path to the Nobel Prize. I would just get Gazzanelli to grow the P. falciparum. I would have to do almost no work whatsoever, because Ricardo would grow the falciparum, then he'd FedEx it to me in the US, and we had all these mice and engineered cell lines, and we could just immediately figure out which TLR was being activated by the parasite, and then we would use some biochemistry to identify the ligand, and then publish the paper in like Nature or the New York Times or something like that. And we would collect our Nobel Prize in medicine, just like Bruce did, right? Except it didn't work out that way. Actually, it turned out that there were lots of problems with it, mostly I won't go into. But somebody had already said, oh, this is silly. We know that malaria is coated with these GPI anchors, okay? And obviously they're the endotoxin of malaria. And sure enough, so this is another cartoon of GPI anchors, and these are very common in parasites. I think all parasites have glycosylphosphatidylmannose anchored proteins on them, and these are pretty immunostimulatory. And this was another easy thing to do, because Ricardo's lab had already purified GPI anchors. And he sent us some purified lipid. Actually, we sent a student to his lab here, and it turned out that TLR2 was the receptor for GPI anchors, and that was great. And Chain Gowda at Penn State subsequently published a paper in JBC showing that GPI anchors from P. falciparum had the same type of activity that the GPI anchors from T. cruzi had. And we thought that was great, except Chain did he actually counted the number of molecules on the parasite and the potency of the GPI anchors, and he concluded that whatever's going on in malaria wasn't really due to GPI anchors, because they weren't potent enough and there wasn't enough of them on the parasite. So there went that chance for a Nobel Prize. But then Shizuo Akira came along. I think Alan knows this story pretty well. Shizuo Akira came along, and Shizuo is, he is an amazingly productive and great person. And I don't want anybody to come away from hearing this story thinking that I don't think he's wonderful, because I think he's a fabulous guy. But he published this paper in a little-known journal about something called hemozoin. Now, what happens in malaria, the parasite loves blood, right? And it metabolizes the hemoglobin from blood. It ends up with hemin, and what it does is it condenses this very toxic hemin molecule into hemozoin, or you can see it in the tissues of people who have died from malaria. They're all stained with malarial pigment. You can see it within the erythrocyte itself. And Shizu has this paper published in, I can't really read this, but it was published in a journal in the United States. And he said TLR9 mediates innate immunity by the malarial pigment hemozoin. And we thought that was pretty cool because we had been studying TLR9 in some depth. And they concluded that hemozoin was the first example of a non-nucleotide ligand for TLR9. And of course, there went, now this seemed like real to me, and I was just not so thrilled that my Nobel Prize seemed to be getting further and further away from me. And then Peggy Peroche, there she is here, Peggy, she started working on this project, and she couldn't reproduce anything that the Akira Lab had published in this great paper that had been published. And so instead of using what they called synthetic hemozoin, which is where you buy the hemin from Sigma and you put it in solution and it forms huge crystals. Actually, the crystals that the synthetic hemozoin forms are about the size of about 1,000 monocytes lined up one after another. If you look at them underneath the microscope, they're tremendously big. You have to sonicate it. And in any case, she found that if she avoided synthetic hemozoin but instead purified it from P. falciparum cultures, that in fact it indeed did activate TLR9. See, wild-type cells are stimulated, the TOL9 knockouts, and the MID88 knockouts. MID88 is an adapter used by TOL9. They weren't activated, and so that part of the story seemed to be real. But when she also found out if she took this natural hemozoin and she treated it with several DNases, that the activity went all away. So it wasn't really the crystal that was stimulating TLR9 here, but it was the DNA that was coating the crystals that was the bioactive ligand. Of course, hemozoin does have activity. I'm not going to go into this, but it's been shown by many groups now that NLRP3 inflammasome is activated by hemozoin, and obviously interleukin-1 beta is an important part of the malarial syndrome. All right, but of course, working with Gazzanelli here, he pays attention to a lot of things, and almost immediately he published this paper in PNAS, and he let me be a co-author on this paper, where he took a drug that was a specific TLR9 inhibitor and showed that it blocked actually death in an experimental animal model of malaria by inhibiting TLR9. It's a beautiful paper, and I recommend it to you all. So now we had a problem in that TLR9 was originally proposed to preferentially bind CpG-rich islands of DNA, and P. falciparum is actually AT-rich. And in fact, it's unbelievably AT-rich, and it contains these stem loops, A followed by 5 or 6 T's, and A and a C, and there are thousands of these. So it's a very, very common motif in the P. falciparum genome. There are over 6,000 of them. It really leads the list for having these AT-rich motifs. And we thought, well, that doesn't sound like a very good ligand for TLR9 because it's not a CpG motif. And enter the lab is Shruti Sharma, and Shruti thought that this was very interesting, and she decided to try to figure out how these AT-rich motifs might be immunostimulatory. And she designed a whole bunch of synthetic oligonucleotides that were based on this stem loop motif and started studying cells. And to make a long story short, what she did is she went back to some earlier work that we did with a fellow shown here, whom Ricardo found. I'm not quite sure how Ricardo found him, but this guy was, and unfortunately he's passed away too, but he was quite a brilliant malariologist. Luis Hildebrando Pereira da Silva. I mean, he was an amazing man, for those of you who didn't get a chance to meet him. He had more energy than any person I've ever met in my life. In any case, we went up to Porto Velho with Ricardo, and we collected patient specimens, and we found, using microarrays, that's how old this is, that P. falciparum drives a type 1 interferon response. So this really interested Shruti, because here we have this AT-rich motif. It's not a classic CpG type of motif. It shouldn't really drive TLR9, and that interested Shruti. So she started looking at the basis of this type 1 interferon motif. And by the way, another Brazilian who happened to work in the lab at the time did this experiment in the PB-ANCA model. This is an experimental cerebral malaria model. And she looked at the interferon alpha-beta receptor knockout and found that, in fact, that animal was highly protected from fatal cerebral disease. So it really looked like there was a good reason to be studying this type 1 interferon response. And to make a long story short, Shruti generated TRIF MYD88 double knockout mice. When you do that, basically your toll receptors are knocked out. Those animals no longer responded to classic CpGs through toll9 because they didn't have the adapter proteins, but they did respond to these AT-rich motifs. So this was a toll receptor independent response that seemed to be happening in malaria. And Shruti went on to show that the basis of this had to do with the activation of a system known as the C-gas sting system. And again, because of time, I'm not going to go through all of this, but you'll hear about it next year because I'm willing to place a bet that the discoverer of C-gas wins the Nobel Prize this year. I think so, James Chen. He's got my vote, even though I'm not on the committee. If I were, I'd start by voting for myself, believe me. But anyway, this is a very important immune pathway. And Shruti discovered that, in fact, that C-gas mutants were hyporesponsive to hemazoin. Okay. Now I'm going to finish up here. I hope I'm within my half hour. I haven't been paying attention to the time. I have 15 minutes. I only have 2 slides. So I just want to say that I actually, I didn't go into this, but when I started studying malaria, what I really wanted to study was subclinical malaria. That is, I discovered once when I was taking a trip in Africa that there were kids who were playing soccer who had 5% parasitemia in their smears. And that was actually one of the first things that attracted me to the disease of malaria. You see this kid running around and you know that he's had a blood smear done and it's 5% parasitemia, which, to put it into perspective, if that happened to me, I would be in the intensive care unit, probably on a respirator in Trendelenburg and the whole thing. So, and actually in highly endemic areas, this is, it's only really little kids who don't have any immunity whatsoever who get very, very severe malaria. It's not adults. So older children and adults typically have subclinical malaria. And that, of course, was something that Gazzanelli also picked up on and decided to study that here in Brazil. In our earliest papers, we thought, well, maybe malaria might activate toll receptors, and we would find that some of the toll receptors were inactive, that they had gotten sort of desensitized because of malaria. And Ricardo had a beautiful paper showing that in fact isn't true, that malaria primes monocytes to have an enhanced response to TLR ligands. That was one of our earlier ideas. But then Ricardo went back and looked at subclinical malaria in the Porto Velho area. And there were a lot of interesting discoveries from that time. Lis was part of that work too. One of which I think is really important is that if you take blood from patients who have subclinical malaria, even if you can't see it on blood smear, even if they're only PCR positive, so it's very, very low levels of parasitemia, you can still infect mosquitoes. So here, with the help of Maísa, is Maísa on this paper? She should be. Maísa da Silva, gotta go back. Yes, Maísa da Silva, I can't pronounce that name, Araújo. With the help of Maísa, we made this discovery. And I think this is important because the world is trying to eradicate malaria right now, right? And people are walking around, they don't know they have the disease, and they can get bitten by a gravid mosquito and transmit the infection on to others. Of course, in Brazil, the level of malaria isn't so high. It isn't a hyperendemic region, but in the truly hyperendemic regions of the world, the amount of subclinical malaria is incredible. And for example, in Kisumu, which is right here, this is Lake Victoria here, the blood bank samples, when tested by PCR, are greater than 25% positive for P. falciparum, meaning that if you need a blood transfusion when you're in Kisumu, you also need to be treated for malaria. And we have recently been studying subclinical malaria in school-age children in a neighboring community called Chilambo, right next to Kisumu. And 25% of the school-age children are blood smear positive in this community, but asymptomatic. And they obviously represent a huge reservoir for P. falciparum and a huge problem for those people who would like to rid the world of malaria, which is a very good goal, in my view. So overcoming the challenges of subclinical malaria is truly one of the major obstacles in eliminating the disease. Hopefully the vaccine will make some inroads to that, but I'm not overly optimistic about that, especially since a lot of the approaches that the world has taken have sort of been disrupted recently by our disruptive president. So, I want to thank Ricardo for introducing me to Brazil, introducing me to malaria, and helping my career along, and Lis, who's been a great collaborator. This is a shout out to Hildebrando, even though he's not with us anymore, we won't forget him, and Delio, who now works up in Porto Velho, Kate Fitzgerald, who's worked with me on all my malaria stuff in vitro and in mice for years, the current members of my laboratory, the collaborators, including Shizua, who's an incredibly great collaborator. He gave everybody all his reagents over the years, has helped out everybody in the entire innate immunity field, and Kensuke, Fabian Kusowski from Eisai, Weidhorning, Eichelotz, Stephanie Vogel, and many, many more, and a few of the truly great younger scientists who have worked in our lab, Warson and Draji, and I see Milton is actually here, and he's an incredibly talented young scientist who we were lucky enough to have join us for a couple of years. So, Ricardo, congratulations, 30 years. You've had a big impact on a lot of people.
Thank you very much. Thank you very much, Doug. Wonderful. Last but not least, I'd like to introduce Professor Jorge Kalil from Faculty of Medicine at the University of São Paulo. Kalil was also the president of the Brazilian Society of Immunology, the president of the International Union of Immunology Societies, the Butantan Institute, so many other things. Again, please go Google, Wikipedia, all these incredible guys. Thank you, Kalil.
Well, thank you, Ricardo, for inviting me. It's a great pleasure, great honor to be here. I mean, you have done so many things in these 30 years. I tried to speak a bit during this video. And you are a true Grazinelli also, and I live with many Grazinellis in my life. When we had these programs in FINEP and CAPES and CNPq and all this, very often I represented biology, and Ramayana Grazinelli was the representative of physics. And we have these boring meetings in Rio. Well, my thing was it was in Rio. And, but the interesting things in those meetings were when early in the morning, Ramayana and myself, we would walk on Botafogo Beach several times, going back and forth and talk about science and development of science in Brazil. And also for at that moment that I knew that there was a guy called Ricardo Gazinelli that was living in the United States, as I mentioned there. So I have a good souvenir from Ramayana. Ricardo has many things that are compared to him. Giovanni Gazinelli also was a very interesting and important Gazinelli. And they have all contributed a lot to science. And they were all not easy persons. In Portuguese, you say ranzinza. But they are very good friends. Frankie? Frankie, okay. Could I return to first slide? So I think I completely messed up what I had to do here, because I'm not going to talk about me, and I'm not going to talk about the development of science. I'm not going to talk about even the development of the science of immunology in vaccines, because actually the real development of the immunology in vaccines is from the last 10 years, I would say. Otherwise, we have used vaccines. So I thought that in this Center for Vaccines and Vaccination, control it here, okay. I'm going to talk about the history of vaccines in Brazil. I received the invitation, so that's why the title of my talk for today was in Portuguese, and the title for tomorrow was in English. So I prepared these slides in Portuguese and the slides for tomorrow in English. But, okay, I'll try to speak in English. I think it will be easy for Alan or Doug to understand what are in these slides. I must say I was not a student of Ricardo. I am much more from the generation of Doug. And let's talk a little bit about it's a beautiful story about vaccination in Brazil. Of course, we have to start saying that this slide is very interesting. See the survival in Breslau in 1690 Anno Domini compared to the Neolithic and Paleolithic. Here, the same life expectancy, survival expectancy. Things started to go a bit better in the year 2000, but nothing compared to what we could find in United Kingdom here. The life expectancy of men in Brazil in the 40s was 42 years old. In 2015, 71. Now it's a bit better. And I'm not going to show the class distinction, but people that are rich in Brazil, the expectancy is 82 to 83. People that are poor is 66, 67 years old. It's a huge difference. But anyway, we had one of the worst number of cases of death in the first year of life, and things are getting much better. I would say that these differences here in the beginning were due to clean water, antibiotics, and vaccines. These are the great differences. Nowadays, there are new stuff that are coming. Exercise, exercise, better eating and things like that. But those are the big differences. Of course, a neurosurgeon is very important with high technology. He may save 100 lives in his life. During his life as a neurosurgeon, he may save 100 lives, not much more than this. But those activities save millions and millions of lives. We all know that Jenner is the one that brought the vaccine. Actually, he didn't invent the vaccine. This was already used in India. And there was a noble woman that came from India and brought the idea to Jenner. So Jenner took the observation was that the cow that have smallpox, people that used to work with those people, those animals, didn't get the smallpox. So the bovine smallpox protected against smallpox, and Jenner started vaccination. So we're talking in the beginning, the end of the 18th century. And this was clear, but there was already a lot of hesitancy on the use of vaccines, even though it was clear for everybody that people were protected for smallpox that killed millions and millions of people. But when you look here, see, we're talking 1,700 and something. In this 20th century, still we had many, many people that died of smallpox. And then the world started doing campaigns. And every time you did a campaign, it dropped the number of smallpox. But smallpox was actually eradicated in 1980 only, actually 1977. So between the discovery of a vaccine that protects against a lethal disease and actually curing the disease, not having the disease, it took 2 centuries. So this problem is an old problem. It didn't come with Robert Kennedy, by the way. It's an old problem. In Brazil, we had the problem as well for smallpox when Oswaldo Cruz, coming from Europe, decided to do a big campaign of smallpox in Rio. And there was this riots against vaccination. And this was 1904. But he was clever enough to create the Oswaldo Cruz Institute that was created 1 year or 2 years before this campaign of vaccination. And the other guy that was tremendously important for Brazil in the vaccination was Vital Brazil, that created Butantan Institute about the same time. At that time, these institutes were more serotherapic institutes, so they used syrup from animals to try to cure those diseases. Butantan is still serotherapeutic. I had to heard once from a very famous Brazilian that lives in the US that we are still very primary in our treatments because we still use serum from horses. The problem is that botrops, here is a big jararaca, botrops. There are at least 10 different venoms, so it's very difficult to treat with monoclonal antibodies, everyone. But those guys created those institutes that were very important in Brazil because they brought in first the serotherapy and then the production of vaccines. And but actually, the first vaccine that was created after research was the ones that were created by Louis Pasteur. So in the middle of the 19th century, Louis Pasteur, he discovered a lot of things. And in 1885, he had attenuated the rabies virus and has treated a boy that was bitten by a rabies dog, his name is Münster, and he developed all the concept of having attenuated virus to do the vaccines. So we're talking Pasteur by the end of the 19th century, and then we had Oswaldo Cruz that was there, that came and brought the idea of having an institute like this in Brazil. And then we had those campaigns to stop vaccination, but we are not really organized. And there was a big campaign in '62, and the last case in the Americas was in 1971. And the last cases were in Somalia, in Africa, in 1977. Ever since we don't have smallpox anymore. We tried to do this with other diseases, but we are unable to do it. We can talk about this. What are the problems that are related? And then we started the oral vaccination against polio, and this is due to Sabin that actually gave his discovery of the attenuated virus to many countries, including Brazil. And Fiocruz still produces the same vaccine from Sabin in incubators that date from the 50s or 60s, but this is one of the best vaccines we have. Just a comment. It's one of the best vaccines we have because there is no big campaigns of yellow fever. Because if there were big campaigns of yellow fever, we would have much more hesitancy than we have with the COVID vaccines. Because 1 in 1 million people dies by using this vaccine. And 1 in 100,000 people have nervous systems problems by using this vaccine. But still the best vaccine. And then I ask you, how can we test a new vaccine against yellow fever? There are many people, when I was president of Butantan, that came to me and said, I have a new vaccine against yellow fever. Think about how we're going to test a new vaccine against yellow fever. I don't have any idea, and that's why we're still using this vaccine that dates for more than 100 years. So Brazil started getting organized when the WHO in 72, 73 said, people, you have to get organized against vaccination. That's the best you can do for the health of your country. And Brazil created the National Program of Immunization. And this was a great achievement. And the idea was to integrate the vaccination and adapt it to different areas of the country. I have to cut a lot of slides, but you know that we can go in the Amazon, deeply in the Amazon, 2 weeks, 3 weeks in ships to go to different human settlements to vaccinate everybody. And the program was very successful. I'm not going to talk about all the program all the time, but just you to have an idea of the scenery, epidemiological scenery at the time of the creation of the program. The number of cases of poliomyelitis, the polio per year, were 11,000. I can see that most people here don't have gray hair like me, but I have gray hair. I have seen people with polio when I was a kid at school. Smallpox, number of cases. Well, at the time of the creation of the national program, we didn't have more, but in the 60s was almost 2,000 cases per year. Diphtheria, 10,000 cases. How do you call it? Tuberculosis, 110,000. Measles, 110,000. When people asked me, have you been vaccinated against measles? Alan, have you been vaccinated against measles? Yeah. No. You got measles. There was no vaccine at that time. So my so my father, that was a pediatrician, so he controlled in the family. When there was a kid with measles, and the guy was in good health, they would put the kids together, so everybody had measles, and he would control. And all this happened for the different infectious diseases that nowadays people that have that don't have gray hair, have been vaccinated. So when I took a position at Harvard as a visiting professor, they asked me for all my vaccines. I said, they don't have vaccines against measles. I said, why? Because I got measles. There was no vaccine at that time. So I had to show by serology that I had been infected with all these viruses. And many people died. But then we started the program, and the program, I'll show you, went up, and we could achieve very high numbers already in the early 80s. And this continued to go up, and then the results were wonderful. So see, polio disappeared, almost disappeared between the 60s and 70s. Although, as I said, when I went to school, at that time, there were still some cases. Measles disappeared. I will talk about measles later. And this is the estimated number of lives that we saved because of the vaccination. It's amazing. Tetanus was also a big problem. And who studied medicine here probably haven't seen a case of tetanus. I have seen several cases when I was a student, because at that time there was no prevention, especially in rural areas. And people would, it was terrible to see. But the number of neonatal tetanus went practically came to zero. Accidental tetanus, we still have some, but the tendency is going to zero because people sometimes don't take the booster shots. That's why we still have some cases. And then with all the programs, and especially here in '86, they created the Zé Gotinha. Zé Gotinha was a very nice personage that appeared in TVs. At that time, all the family after dinner would go to watch TV together. Remember that? It doesn't exist anymore, right? First, the families don't have dinner together. Second, the young people don't watch TV anymore. They do social media. And I'll return to this because it's very important what I'm telling you by the way we communicate about vaccination. And I have been discussing this for many years at the Ministry of Health. So at that time, everybody would go and watch TV, and there was also Zé Gotinha. Do you remember who watched TV at the 80s? And so this was '86. And we created a very good program of education on vaccines. And Brazil perhaps was one of the countries that was really friend of vaccination. So when we reached 1998, here in this period between 95 and 2000, we had improved a lot the production of vaccines in Brazil. I'll return to that. I was a part of it. And then we reached a plateau around 2000. And the levels of vaccination were between 95 and 100% of the target population every year. So we could immunize really almost everybody. And this continued on till 2015, 16, by the time, by the time of Dilma government, just for you to have an idea on the timing, it starts declining. Why does it start declining? There are many reasons. When I asked my students, I have students that are doing post-graduation studies in immunology, and I asked them, did you vaccinate your kids? And many of them didn't. I said, I can't believe that you did not vaccinate your kids. And they say, well, you know, why I would vaccinate against measles? Well, said, measles is a terrible disease, it can be fatal. Yeah, but I have not seen a case of measles in my life. Nobody has measles anymore. And this story that it can cause a problem, autism, things like that. But, I mean, it has been shown that's not true. It said, well, but where is this smoke, there's some fire, right? With the same Brazil. So people don't vaccinate, many people don't vaccinate. Well-educated people don't vaccinate because one of the reasons, these diseases are not there anymore. That means we don't see. So they say it does not exist. Second, there is no zagotin anymore. We don't see the publicity that used to be in TV. Why? I mean, the families don't go to see TV and the government start doing the program. So we discuss a lot at the Ministry of, at the National Program of Immunization, how we can reach young people to get immunized. And third, that there is this anti-vax. All the fake news anti-vax that are very strong in US and France. And it's very difficult to fight because when you see at the internet people talking against vaccines, they show very big stuff, you know, that impress you a lot. When a boring scientist talks for vaccines, he's boring. He's boring. He also took my wife keeps saying to me, I don't know why they call you in TV, you're boring. You tell just the truth. I said, well, that's the way we are as scientists. But this is a problem to fight. And that's why I'm bringing to you. You see, I'm a kind of a priest here. So, and then Brazil, see, look at these numbers here. Here, there are much less numbers of people that have been vaccinated. And then in Venezuela, we have this great guy, Chavez, that didn't have money not even to vaccinate his population. So people didn't get vaccinated in Venezuela, with Maduro neither, and they didn't have also jobs. So they invaded Venezuela and they entered Brazil through the Amazon. And they went to Manaus. Brazil was free of measles since 2010. And then in 2014, 15, those Venezuelans started to come. And in 2018, we had an outbreak of measles in Brazil. 1 month after the first case in Manaus, there were already cases in Rio Grande do Sul. See, Brazil was free, and then Brazil was one of the top 10 in number of measles because of the vaccine hesitancy in the country, so about 10,000 cases. And of course, it worried us a lot, people that were involved in vaccination. So when you look at the number of cases in Brazil, it's clear. See, 2018, 19, the number of cases was enormous, and we start a big campaign again. But not everybody wanted to be vaccinated because of this guy. I mean, this guy, you know the story, right, of Andrew Wakefield that started the anti-vaccine movement, saying that measles would induce autism. So all the numbers and the cases were reviewed, and the paper was retracted. This guy lost his title of medical doctor, but the damage was done. And then there was, Alan perhaps knows it better than me, there was this important person in the United States that was a Playboy rabbit that went to the TV and said that her daughter was autistic because of vaccination. And of course that she's more truthful than any scientist, so everybody started again not vaccinating. So this is a big problem that we have to face. And we had this surge of yellow fever in Brazil, 2017. Do you remember that? So now Brazil was pretty well organized. Not everybody in Brazil is exposed to yellow fever, so not everybody is vaccinated against yellow fever. But the guys that follow, and this is very well done, this epidemiology, that follow the monkeys in the forest, they start watching that people from the monkeys from the Amazon start dying of yellow fever, and this went down by the forest and went to São Paulo. And they could predict that when they would see monkeys, that in 1 or 2 weeks they will start having human. The yellow fever here was not the yellow fever, city yellow fever. That is another transmitter. It became only in the forest. But the people could preview that yellow fever would reach that particular area, and we started in Brazil, in São Paulo, to vaccinate everybody that were in this region. But not everybody was vaccinated. Not everybody believed. But there was one, that's very interesting, there was one city in this row where the mayor was a physician. And she almost obliged everyone to be vaccinated against yellow fever. And when you do the epidemiology, it's beautiful. It's too bad that I don't have the slide here, because you have yellow fever coming down, and then there is an opening that is this town, and then it continues again. So people were not vaccinated. And who was not vaccinated? It was mainly men between 20 and 50 years old. So those are the men that enter the jungle to work, the forest. And those are the ones that are prone to have disease. But of course that a man is not going to be defeated by a stupid virus or a mosquito. So they were macho men, they were not vaccinated, and they were dead. So we lost a lot of people in this this yellow fever story. And then the Brazilian government say, okay, we have to vaccinate everybody in the country against yellow fever. That's all stupid, because people in the Northeast there is no yellow fever. People don't move that much. And I mentioned to you, 1 dies every 1 million people. So if you vaccinate 10 million, 10 will die. In a city of 5 million, 5 will die. And this bring again the anti-vaccine thinking. See, I mean, people have to be clever and think about the consequences, whatever they do. So what seemed to be, it was an engineer, I'm not going to tell the names, but the guy that decided this was an engineer that was Minister of Health. I talked to him and I said, this is not a good idea. He said, but people cannot blame me of having yellow fever. I said, but not in this region, not in this region. So these are the numbers of yellow fever in 2017, 2018. Of course, we can have it again. What we need is to have the vaccine ready to go. And then in 1991, finally they realized after 5 minutes, I'm talking, okay, I'm sorry. I will try to be quick. They thought that it would be important to have a technical committee, the same one that was, that everybody had to resign this week by Kennedy. There's no more in CDC, this committee. And we created a group of people that would discuss what would be the best policies for immunization. We started then to vaccinate other not only children, but also influenza. By the way, the influenza vaccine in Brazil is done by Butantan. I was the one that launched it. And there was a program of national production of the vaccines that are the most used in Brazil. Not all are completely produced in Brazil, but they are at least finished in Brazil. And we had this case of problems with meningitis C. I don't know if you remember, in the region of, in Campinas, it was around 95, 96. And and Professor Jatene was the one that was in charge of the Minister of Health, and we brought a lot of experts to see what the problem was, and the problem was the good manufacture practice. And then we started a program, and I was at the early beginning of this, 1995, 96, when Jatene was the Minister of Health, to improve the production of vaccine in the different institutes in Brazil. Good manufacture practice, so we started in 95, 96 in Brazil. We have a calendar of, national calendar of vaccination. I'm going to hurry up. And we have these centers of references for microbiological products that not only vaccinate, but can use also anti-sera, monoclonal antibodies, and so on. This is the national calendar of vaccination for children, teenagers, adults, and pregnant women. And the main results was that we eliminated many diseases. Have to hurry up. We have in Brazil nowadays 49 products that are being delivered by the SUS, by the National Program of Health. We have 38,000 rooms for vaccination in the country with people that have technical people that have been trained, and 52 centers of reference of immunobiologicals that are more sophisticated. For instance, someone says, I cannot receive the influenza vaccine because I'm allergic to eggs. So there's no problem to vaccinate, by the way, because I cannot go in detail on the allergy, but these people go to these centers where they can be vaccinated and evaluated every time. The problems that we have, why we have less numbers in vaccination, may be related to many things, as I mentioned to you, and the negationists and the fake news is the most important. The suspension of the technical cameras, they came back, the chambers, for discussions. There is not a good communication between the states and the cities, and so on. So we are fighting all this. So we want to keep the national program going on and offer the Brazilians with all the vaccines that are suggested by WHO, and that's the way to go. And Brazil is recognized as perhaps the best public vaccination program that exists in the world. And we should keep it. So I ask you to help us to continue. And for this, vaccinate save lives. So vaccinate yourself and your children, your family. Don't show vaccines with a syringe. People are afraid of syringes. Every time they talk about a vaccination, what they show? A syringe. When they talk about monoclonal antibodies, do they show a syringe? No. Any others? So don't show syringes anymore. Avoid syringes. People don't go there because of syringes. I'm serious. See, you have noticed that there's no syringe in my slide. Huh? Well, I thank you. Thank you very much.
Thank you very much, Kalil, for this very profound, inspiring talk. I think we now have closed the session. Yeah, I mean, anybody has questions to any of the speakers? Otherwise, we go for a break, coffee. Okay? And then come back in 15 minutes. Thank you very much.